Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to be covering Chapter 3 in our MCAT Biochemistry series, and this chapter is titled Non-Enzymatic Protein Function and Protein Analysis. In this chapter, we're going to be covering the following objectives. First, we're going to talk about non-enzymatic protein functions inside the cell. That includes structural proteins, motor proteins, binding proteins, cell adhesion molecules, and immunoglobulins. Next, we're going to look at biosignaling. This section covers how proteins help transmit signals in the body using mechanisms like ion channels, enzyme-linked receptors, and G-protein-coupled receptors. Then we're going to move on to protein isolation techniques. We're going to go over two major methods, electrophoresis and chromatography, and learn how scientists separate proteins from complex mixtures. Finally, we're going to discuss protein analysis. Here, we're going to explore how researchers determine a protein structure, amino acid composition, functional activity, and concentration. So with that introduction, let's go ahead and get started with our first topic, cellular functions. We're going to begin with covering structural proteins. These proteins help give the cell its shape, and they provide mechanical support. They make up the cytoskeleton, they help anchor proteins in place, and they are found throughout the extracellular matrix. The most common structural proteins are collagen, elastin, keratin, actin, and tubulin. Let's go over each. Collagen is a triple helix fiber, and it makes up most of the extracellular matrix in connective tissue. Elastin also contributes to connective tissue, and it helps it stretch and recoil. Keratins are intermediate filament proteins that are found in epithelial cells like skin and hair. Actin forms microfilaments, and it's also found in the thin filaments of muscle. And finally, tubulin builds microtubules, which are involved in cell structure and transport. So that's structural proteins. Let's cover motor proteins next. Motor proteins are responsible for movement within the cell, and they work by undergoing conformational changes that are powered by ATP. So in other words, they act as ATPases to convert chemical energy into mechanical work. Myosin is the main motor protein that interacts with actin, and it's involved in muscle contraction. Kinesins, they play a role in aligning chromosomes during metaphase and depolymerizing microtubules during anaphase of mitosis. So in other words, they help move chromosomes during cell division, and they also transport cargo along microtubules. Then there's dyneins. These are involved in the movement of cilia and flagella, which help some cells move or sweep substances across surfaces. Each of these motor proteins has specialized roles, but they all help move things around by walking along the cytoskeleton structures that are inside the cell. Then we have binding proteins. These proteins bind to a specific target, like a hormone, an ion, or a molecule, and they either hold onto it to keep it in place, or they help regulate its concentration. The key point is that they stabilize or transport molecules without modifying them. Next are cell adhesion molecules, or CAMs. These proteins allow cells to stick to each other or to the surfaces in their environment. They're very important for tissue formation and immune responses. Now, there are three major types of cell adhesion molecules. The first type is cadherins. These are proteins that help cells stick to other similar cells, and they need calcium to function properly, which is why they're called calcium dependent. Cadherins are especially important in holding cells together in tissues like skin or the linings of organs, and they help those tissues stay organized and intact. The stronger the cadherin interactions, the more tightly the cells are held together. Then there's integrins. These are proteins that span all the way across the cell membrane. You have one end that sticks out of the cell and it attaches to proteins in the extracellular matrix. This is the network of proteins that are outside the cell that gives tissue structure. And then the other end connects to structures that are inside the cell and it helps anchor the cell in place. In addition to keeping cells in place, integrins also help cells sense signals from their surroundings and respond, like when to know that they need to move or grow. Finally, there's selectins. These help cells stick to carbohydrates on the surface of other cells. Now, these interactions are weaker and more temporary than cadherins or integrins, which is important for processes like immune cell movement. So, for example, when there's inflammation or there's an infection, selectins are going to help immune cells slow down and roll along blood vessel walls so that they can exit the bloodstream and then move into the affected tissue. Now let's talk about antibodies. These are also called immunoglobulins or Ig. These are Y-shaped proteins that are produced by B cells. This is a type of white blood cell that's involved in the immune response. Antibodies are made in response to antigens, which are foreign molecules that are found on the surfaces of viruses, bacteria, or toxins. And the main role of an antibody is to bind to an antigen and either block its harmful effects or mark it so that other immune cells can remove it from the body. Now, each antibody is built from 4 protein chains. There are 2 identical heavy chains and 2 identical light chains. And these chains are held together by disulfide bonds and by other non-covalent interactions that stabilize the structure. The antibody further has 2 major regions. At the tips of the Y shape, there are variable regions on both the light and heavy chains. And these variable regions form the antigen binding sites. They are called variable because their structure differs between antibodies, which allows the immune system to produce a large variety of antibodies, each one that's specific to a different antigen. The rest of the antibody is known as the constant region. So this part of the structure is more similar across antibodies of the same type. And the constant region, it doesn't interact with antigens directly. Instead, what it does is it determines how the antibody will signal or interact with other parts of the immune system, such as immune cells or complement proteins. Now, because antibodies have one part that recognizes the threat and another part that activates the immune response, they're highly effective at helping the body detect and eliminate harmful invaders. With that, we can move into our second objective, where we're going to discuss biosignaling. Biosignaling is the process by which cells receive and respond to signals from their environment. This process is essential for communication between cells and for coordinating physiological functions throughout the body. One important class of biosignaling proteins is ion channels. These are membrane proteins that form specific pathways for charged molecules like sodium or potassium ions to pass through. They allow for facilitated diffusion. This means that ions move down their concentration gradient without using energy. There are 3 main types of ion channels. We have ungated channels. These are always open. They have no gates, so ions can move freely through them depending only on the concentration gradient. Then we have voltage-gated channels. These open or close in response to changes in membrane potential, and they are especially important in neurons and muscle cells. Finally, we have ligand-gated channels. These open or close when a specific molecule, a ligand, binds to the channel. And that ligand might be a neurotransmitter, it might be a hormone, or it might be some other signaling molecule. Then, in addition to ion channels, some membrane proteins act as enzyme-linked receptors. These receptors respond to ligand binding by triggering a cascade of reactions inside the cell. These enzyme-linked receptors have three key parts. The first part is a membrane-spanning domain, which anchors the receptor in the cell membrane. The second part is a ligand binding domain, which is where the signaling molecule is going to bind. And the third part is a catalytic domain, which becomes activated and it carries out an enzymatic function inside the cell. Now, these types of receptors, they're involved in a lot of signaling pathways, especially those that are related to growth, metabolism, and immune responses. Finally, there are G protein coupled receptors, or GPCRs. These are a large family of membrane proteins that are involved in signal transduction. So when a ligand binds to a GPCR, it triggers a cascade of events inside the cell by activating a G protein. And there are three main types of G proteins that we need to know. The first is GS. This stimulates the enzyme adenylate cyclase, which increases the amount of cyclic AMP inside the cell. Then there's GI. This does the opposite. It inhibits adenylate cyclase, which decreases the levels of cyclic AMP. Finally, there's GQ. It activates phospholipase C, which cleaves a membrane phospholipid called PIP2 into DAG and IP3. IP3 then opens calcium channels in the endoplasmic reticulum, and that's going to help increase calcium levels inside the cell. All G proteins are made of three subunits: alpha, beta, and gamma. When inactive, the alpha subunit is going to be bound to GDP. But once the receptor is activated, GDP is replaced with GTP. And the alpha subunit is going to separate from the beta and gamma subunits to either activate or inhibit a target enzyme, like adenylate cyclase or phospholipase C. And these signaling pathways, they're widely used in hormone signaling, in neurotransmission, and in sensory pathways like vision and smell. With that, we've covered objective 2. And we can move into objective 3, which is about protein isolation. One of the most common ways to isolate proteins in the lab is through a technique called electrophoresis. Electrophoresis uses a gel matrix to observe the movement or migration of proteins when an electric field is applied. So here's the idea. Proteins have charge, and when you place them in a gel and apply a voltage across it, the proteins are going to migrate through the gel towards the electrode with opposite charge. So the gel acts like a molecular sieve, and the speed at which each protein moves depends on three things. 1, the strength of the electric field. 2, the net charge on the protein. And 3, the frictional resistance it experiences as it moves through the gel. And this relationship is captured by the equation you see here. V equals E times Z divided by F, where V is the velocity or speed of migration, E is the electric field strength, Z is the net charge of the molecule, and F is the frictional coefficient, which depends on the size and shape of the protein. Now, the most common gel that's used in this method is polyacrylamide gel, which forms a dense cross-linked matrix that separates proteins based on size and charge. There are 3 types of electrophoresis you should know for the MCAT. The first is native page or native polyacrylamide gel electrophoresis. This keeps the protein in its native folded state. That means the protein's charge and shape are preserved, but because different proteins have different mass to charge ratios, the results can be a little hard to interpret with this method. Still, it's useful when you want to study protein function or interactions in their natural form. The next type is SDS page. This is a variation that denatures proteins and coats them in a negatively charged detergent called SDS. What this does is it masks their native charge and it eliminates differences in shape, so the proteins are separated purely based on size. Smaller proteins are going to move faster through the gel, while larger ones are going to move a lot more slower. However, because the proteins are denatured, you can't recover their original functional form afterwards. The third technique is isoelectric focusing. This method separates proteins based on their isoelectric point, or pI. The isoelectric point is the pH at which a protein has no net charge. So in this setup, proteins are going to be placed in a gel with a pH gradient and an electric field. And each protein is going to migrate until it reaches the part of the gel where the pH equals its pI. And at that point, it stops moving because it has no net charge. And by the way, to make a protein move during electrophoresis, the pH needs to be different from its isoelectric point. I really want to emphasize that. Then at its pI, the protein won't migrate because it has no net charge. Each of these techniques gives you different kind of information. So native page helps study function, SDS page helps compare sizes, and isoelectric focusing helps identify the pI of the protein. Now let's talk about another important technique that's used in protein isolation, and that is chromatography. Chromatography is a method that separates and helps identify compounds based on their physical and chemical properties, and it's especially useful when we're dealing with pretty complex mixtures. The key idea is this: if a compound is similar to its surroundings, it's going to interact more with them and it's going to move more slowly. However, if it's less similar, it's going to move more quickly. So based on how fast or slow different molecules travel, we can separate them out. Now, all chromatography techniques rely on two phases. The first phase is the stationary phase. This is typically a polar solid that stays in place. And the second phase is the mobile phase. This is a liquid or gas that moves through the stationary phase and carries the sample with it. As the mobile phase flows through the stationary phase, compounds are going to interact with both phases to varying degrees. And the result is that some compounds move faster and others move more slowly. If a compound has a greater affinity for the stationary phase, it's going to stick to it more and it's going to take longer to travel. But on the other hand, compounds that have more affinity for the mobile phase, they are going to move more quickly and they're going to elute earlier. And this differential movement is what causes the mixture to separate, and we call this process partitioning. To understand chromatography more, we're going to look at a couple of different types, starting with thin layer and paper chromatography. Both of these are simple and visual methods that are used to separate small amounts of compound. And they follow the same basic principle of chromatography that we just discussed, which is separation based on how strongly each compound interacts with the stationary versus the mobile phase. Now, in thin layer chromatography, or TLC, the stationary phase is a thin layer of silica gel or alumina that's attached to an inert plate, like glass or plastic. In paper chromatography, the stationary phase is simply paper, which is made of cellulose. In both cases, the stationary phase is polar. The mobile phase is going to be a non-polar solvent, and it moves up the plate or paper through capillary action. So it pulls the solvent upward against gravity. And here's how it works. You're going to apply a small dot on your sample near the bottom of the plate. This is called spotting. Then you place the bottom edge into a solvent. And as the solvent rises, it's going to carry the different components of the sample along with it. Compounds that are more nonpolar travel further because they interact more with the mobile phase. But more polar compounds, those are going to stick to the polar stationary phase, and they're going to move less. Once the solvent has moved far enough, you're going to remove the plate, and you're going to measure the distance that each spot traveled. You can calculate then something called the RF value. This is the ratio of how far the compound moved to how far the solvent front moved. So RF equals the distance moved by the spot divided by the distance that the solvent front has moved. And this number helps you compare compounds and identify unknowns based on how far they travel in a given solvent. I want to reiterate, RF equals the distance moved by the spot divided by distance moved by the solvent front. And this equation also reappears in our MCAT organic chemistry playlist. So it's important. With that, we're going to move into column chromatography next. In this technique, we use a vertical column that's filled with silica or aluminum beads. And these beads make up the stationary phase, and they provide a lot of surface area for compounds to interact with. The mobile phase is typically a nonpolar solvent, and it flows through the column by gravity or pressure. So as your sample moves down this column, different components are going to interact differently with this stationary phase, depending on their polarity, size, or affinity. The more a compound interacts with the stationary phase, the slower it moves through it. The less it interacts, the faster it moves through it and elutes out. Now, this setup really allows you to separate complex mixtures and collect each component as it comes off the column. Let's break down now the main types of column chromatography that you need to know for the MCAT. The first type is ion exchange chromatography. This uses beads that are coated with charged substances. And these beads are going to bind compounds with opposite charge. So if the beads are negatively charged, they're going to bind positively charged proteins or molecules. Then there's size exclusion chromatography. This works based on size. The beads are going to have small pores, so small molecules are going to enter the pores and get slowed down, while larger molecules are going to bypass the pores and move faster through the column. Finally, there's affinity chromatography. This is highly specific. So the beads are going to be coated with some sort of receptor or enzyme or antibody that binds to a particular target compound. And this causes only your protein of interest to stick to the column while everything else washes out. And then you can use a special solution to release the bound protein of interest later. So it really helps you separate what you don't want from what you want. And each of these methods allows you to isolate proteins based on different properties, which makes column chromatography very versatile. Now, to wrap up our chromatography section, I'm going to cover two more advanced techniques very briefly: gas chromatography and high-performance liquid chromatography. Let's start with gas chromatography. This method is used to separate vaporized compounds. So in other words, samples that can be converted into the gas phase. And here what's going to happen is that the separation is based on how well each compound adheres to the absorbent in the column. So in GC, the stationary phase is a coil inside the column, and it's usually made of crushed metal or a polymer. The mobile phase is going to be a non-reactive inert gas like helium or nitrogen. Your sample, first and foremost, is going to be injected into the system, vaporized and carried by the gas through the column. As the sample passes through, different components are going to interact with the stationary phase to different degrees. Compounds that adhere strongly to the column are going to take longer to pass through, while those that interact less are going to move way faster and they're going to elute first. Then at the end, you get a graph as your output, and this graph shows peaks that correspond to the different components based on how long they took to come out. This is called retention time. So that's what you need to know about gas chromatography. Finally, we have high performance liquid chromatography, or HPLC. This works similarly to column chromatography, but it's going to be computer controlled and much more precise. It uses carefully controlled solvent mixtures and temperature gradients, which allows for better separation and detection. HPLC is especially useful when your sample size is small or when subtle physical forces like capillary action could interfere with the results. So it's a preferred method in analytical labs because accuracy and reproducibility are really essential. So with that, we've completed our third objective, and we can move into our last and final objective on protein analysis. Once we've isolated a protein, the next step is to analyze it. Protein analysis helps us figure out the protein structure, its amino acid composition, how active it is, and how much of it we have. So let's walk through each of these one by one. First, let's talk about protein structure. To determine what a protein looks like in 3 dimensions, scientists often use either X-ray crystallography or NMR spectroscopy. X-ray crystallography involves crystallizing the protein and then analyzing how X-rays scatter through the crystal. This gives us a high-resolution picture of the protein structure. But this technique is pretty difficult, especially the step where you are attempting to crystallize the protein. Some proteins are difficult to crystallize. Then there's NMR spectroscopy, or nuclear magnetic resonance spectroscopy. This is used for smaller proteins, and it relies on magnetic properties of certain atomic nuclei to reveal structured details in solution. Second, we can look at amino acid composition. If you want to know which amino acids are present and in what amounts, we can use simple hydrolysis, which breaks the protein down into individual amino acids. But if we want exact sequence of amino acids, we need a more precise method like the Edman degradation technique. This technique removes and identifies one amino acid at a time from the N-terminus, and that allows us to determine the sequence amino acid by amino acid. And this is a fine technique for peptides that are about 50 to 70 amino acids. Third, we can assess enzyme activity. This is especially important if the protein is catalytic. So to measure activity, we follow the progress of a known reaction that the enzyme catalyzes. Often this reaction leads to a color change, which we can track over time. And the rate of that change tells us how active the enzyme is. Finally, we need to determine protein concentration. That is, how much protein is actually present in the sample. This is usually done through spectroscopic methods. One option is UV or ultraviolet spectroscopy, and that takes advantage of the fact that many proteins contain aromatic amino acids that absorb light in the UV range. And while it's useful, this method can sometimes be affected by contaminants that also absorb UV light. So sometimes to avoid this, researchers can use colorimetric assays. These rely on specific chemical reactions between the protein and a reagent that produces a visible color change. And the most common methods are BCA, Lori reagent, and the Bradford protein assay. Let's actually briefly discuss the Bradford assay and how it works. It uses a blue dye called brilliant blue, which is normally a brownish color when it's protonated. When this dye binds to certain amino acid groups in the protein, it loses protons and then it shifts to a blue form. As the amount of protein in the solution increases, more dye binds and the solution becomes more intensely blue. So higher protein concentrations lead to deeper blue color. One important thing to note in this assay is that it can be limited by the presence of detergents or high concentrations of buffer, which can then interfere with the dye's ability to bind properly. Regardless, all three of these assays work on the same basic principle. The more protein there is, the more color change you get. And this allows researchers to construct a standard curve and then determine protein concentration based on how strong the color is. These measurements are super essential for ensuring that the right amount of protein is used in downstream experiments. With that, we've completed this chapter. I hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.